Chapter 3 Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker in 1969, wherein he suggested the 5 kingdom classification based Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of plant kingdom has changed over time. Fungi and the members of Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us also look at the classification within angiosperms to understand some of the concerns that influence the classification systems. The earliest system of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were mainly based on vegetative characters or on the androecium structure systems given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial systems gave equal weightage to vegetative and sexual characteristics, which is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification systems developed, which were based on natural affinities among the organisms and consider not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plant was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data that are then processed. In this way, each character is given equal importance and at the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constituent of the plant to resolve confusions, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stone, soils, and wood. Some of them also occur in association with fungi, lichen, and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and filamentous form like Eulothrix and Spirogera. Figure 3.1. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of the two gametes. These gametes can also be flagellated and similar in size such as Eurothrix, or non-flagellated, non-motile, but similar in size such as Pyrogera. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in species of Eudorina, is termed as an isogamous. Fusion between one large non-motile static female gamete and a small motile male gamete is termed as oogamous. Example, volvox and fucus. Algae are useful to man in a variety of ways. At least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of the food cycles of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, water-holding substances, example algine brown algae and cara-green red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparations of ice cream and jellies. Chlorella, a unicellular algae rich in proteins used as food supplement even by the space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. 3.1.1 Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and outer layer of pectose. Vegetative reproduction usually takes place by fragmentation. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Pyrogera, and Cara. 3.1.2 Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms ectocarpus to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algine. The protoplast contains, in addition to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has the stalk, the stripe, and leaf-like photosynthetic organ, the frond. The vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the Oogonium oogamous species. The gametes are pyriform, pear-shaped and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum and Fucus. 3.1.3 Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of red pigment R-phycoerythrin in their body. Majority of the red algae are marine with great concentration found in the warmer areas. They occur in both well-lighted regions close to the surface of the water and also at great depths in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, Glaciolaria and Gelidium. Table 3.1 Divisions of algae and their main characteristics. Chlorophyceae, common name green algae, major pigment chlorophyll A, B, food stored starch, cell wall, cellulose, flagellar number and position of intersections 2 to 8 equal and apical. Habitat freshwater, brackish water and salt water. Phaeophyceae, common name brown algae. Major pigments chlorophyll A, C, fucoxanthin. Food stored mannitol, laminarin. Cell wall, cellulose and algae. Flagellar number and position of insertions 2 unequal lateral. Habitat freshwater, rare brackish water, salt water. Rhodophyceae, common name red algae. Major pigments chlorophyll A, D, phycoerythrin. Stored food Floridian starch. Cell wall, cellulose, pectin and polysulfate esters. Flagellar number and position of insertion absent. Habitat freshwater, some brackish water, salt water most. 3.2 Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in the soil but are dependent on the water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession or bare rocks or soil. The plant body of bryophytes is more differentiated than that of the algae. It is thallus-like and prostrate or erect that attach to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem or leaves. They may possess root-like, leaf-like or stem-like structures. The main plant body of the bryophytes is haploid. It produces gametes, hence it is called a gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate anthrazoids. The female sex organ called archegonium is flask-shaped and produces a single egg. The anthrazoids are released into the water where they come in contact with archegonium. An anthrazoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free-living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reductional division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds and other animals. Species of Sphagnum, a moss, provide peat that have been long used as fuel and as a packing material for transshipment of living materials because of their capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rocks and hence of great ecological importance. They decompose rocks, making the substrate suitable for the growth of the higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverwoods and mosses. 3.2.1 Liverwoods. The liverwoods grow usually in moist shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwood is thalloid, example Merchantia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverwoods takes place by the fragmentation of thalli or by the formation of specialized structure called gamete. Singular gamma. Gamete are green multicellular asexual buds which develop in small receptacles called gamma cups located on the thalli. The gamma become detached from the plant body and germinate into new individual forms. During sexual reproduction, male and the female sex organs are produced either on the same or different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. 3.2.2 Mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. These stage bear the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverwoods. The capsule contains spores. Spores are formed after the meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. 3.3 Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in Chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte which differentiated into true root, stem and leaves. These organs possess well-differentiated vascular tissues. The leaves in Pteridophyta are small microphylla as in Selaginella or large microphylla as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structure called as strobili or cones. Selaginella and equisetum. The sporangia produces spore by meiosis in spore mother cells. The spore germinate to give rise to inconspicuous small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Some of this specific restricted requirement and the need for the water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transport of anthrazoids. The male gametes release from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte which is the dominant phase of pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds such as plants are called as homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on parent sporophytes for variable periods. The development of zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit considered an important step in evolution. The pteridophytes are further classified into four classes: Coelopsida, that is Coelotum, Lycopsida, Selaginella, Lycopodium, Spenopsida, Equisetum, Pteropsida, Dryopteris, Pteris and Adiatum. 3.4 Gymnosperms. The gymnosperms, gymnos means naked, sperma means seeds, are the plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, they are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally taproots. Roots in some genera have fungal association in the form of mycorrhiza pinus, while in some others, cycas, small specialized roots called colloid roots are associated with nitrogen-fixing cyanobacteria. The stems are unbranched, cycas, or branched pinus cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls which are arranged spirally along an axis to form lacs or compact strobili or cones. The strobili-bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called microsporangiate or female strobili. The male or female cones or strobili may be borne on the same tree pinus. However, in cycas, male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus. The nucleus is protected by envelopes and the composite structure is called an ovule. The ovules are borne on megasporophylls which may be clustered to form female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes in gymnosperms, the male and the female gametophytes do not have any independent free-living existence. They remain within the sporangia, retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards the archegonia in the ovules and discharges their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and ovules into seeds. The seeds are not covered. 3.5 Angiosperms. Unlike the gymnosperms where the ovules are naked, in the angiosperms of flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are exceptionally large group of plants occurring in wide range of habitats. They range in different size from the smallest wolfia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines and several other commercially important products. They are divided into 2 classes, the dicotyledons and the monocotyledons. Summary. Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Algae are chlorophyll-bearing simple, thalloid, autotrophic and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into 3 classes, namely Chlorophyceae, Phaeophyceae and Rhodophyceae. Algae are usually reproduced vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on the water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axis bearing spirally arranged leaves. The main body of bryophyte is gamete producing and is called a gametophyte. It bears the male sex organs called antheridia and female sex organ called archegonia. The male and the female gametes produced fuse to form zygotes, which produce a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant is a sporophyte which is differentiated into true roots, stem and leaves. These organs possess well-differentiated vascular tissues. The sporophytes bear sporangia which produce spores. These spores germinate to form gametophytes and require cool, damp places to grow. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of male gametes to archegonium where gamete is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which the ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed and therefore these plants are called naked seeded plants. The gymnosperms produce microspores and megaspores which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls and megasporophylls are arranged spirally on the axis to form male and female cones respectively. The pollen grain germinates and pollen tube releases the male gamete into ovule where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovules into the seeds. The angiosperms are divided into two classes, the dicotyledons and monocotyledons.